Hello dear students. Today we will study about the cell cycle. So let's begin. Cell cycle is divided into different phases. Majorly it is known as interphase and division phase. Interphase is again divided into three phases: G1, S, G2, and division phase is also known as M phase. That means mitosis phase. G1 phase, as you can see, is about 6 to 12 hours. It prepares the cell for the next upcoming event known as DNA replication in the S phase. S phase is about 6 to 8 hours. Main event that is occurring during this phase is DNA replication. Next event is G2 phase. It is about 3 to 4 hours. G2 phase check whether the DNA has been replicated properly or not and whether the cell is ready to move for the division phase. M phase is about 1 hour. There are certain checkpoints also in the cell cycle. Like as you can see, during G1 phase, the first checkpoint, cell check that whether the cell size is sufficiently large or not. Nutrient conditions are sufficient or not, growth factors are present or not, and whether the DNA replication has been repaired or not. If any of these requirements are does not met, then cell can go away from the cell cycle into permanent resting stage. This checkpoint is known as G1 checkpoint. Then next checkpoint is at the G2 phase. In this checkpoint, cell check whether the cell size is sufficiently large or not, and then whether the DNA replication has been properly completed or not. That means if any error occurred during the DNA replication, that error has been removed or not. If any of these two requirements does not met, then cell can go into permanent resting stage out of this cell cycle. If everything is all right, then cell goes towards the division phase. During division phase, there is one more checkpoint when all the chromosomes are properly attached to the spindle fibers or not, it is checked. These three checkpoints are discovered by Hartwell. Paul Nurse discovered about the cyclin-dependent kinase. Tim Hunt discovered about the cyclin. Together, all three were given Nobel Prize for their discovery of the key regulators of the cell cycle. This is about the molecular basis of the cell cycle. There are two main enzymes which are responsible for the molecular level regulation of the cell cycle. These two enzymes are cyclin-dependent kinase. They add the phosphorus to different enzymes and thereby activate or inactivate the respective enzyme, and they are regulated by cyclin. Cyclin are regulatory unit and cyclin-dependent kinase are catalytic unit. Together, they are known as cyclin CDK complex. In different model systems, different cyclin and cyclin-dependent kinase have been identified. In Saccharomyces cerevisiae, which is a yeast, the CDK is known as CDC28 and cyclin are known as CLN1, CLN2, CLN3, CLN4, CLN5, CLN6, and CLB, which acts during M phase. In Saccharomyces pombe, which is a haploid yeast, the CDK is known as CDC2 and cyclin is known as SIG. In mammalian system, 4 different types of cyclin-dependent kinase and 4 different class of cyclin have been identified. This is the current model of the cell cycle. In yeast, main kinase is CDC28, as we have seen in the previous slide, and different CLN series are working during G1 as G2 phase. During M phase, CLB cyclin is working, as we have seen in the previous slide. Different series during interphase, and CLB during M phase. In mammalian system, CDK is the main kinase, and different cyclins are worked during different phases, like G1 phase cyclin D, S phase cyclin E, G2 phase cyclin A, and M phase cyclin B is working. Thank you very much.